In this video, we're going to discuss the citric acid cycle. So first, let's talk a little bit about the three stages of respiration. Our first stage would include glycolysis and also the breakdown of some other molecules such as amino acids and fatty acids in order to produce acetyl-CoA. Of course, glycolysis produces pyruvate, which then is converted into acetyl-CoA by the pyruvate dehydrogenase complex. Our second stage is where acetyl-CoA enters into the citric acid cycle. As acetyl-CoA enters into this or goes through the citric acid cycle, it is progressively oxidized, producing carbon dioxide and also reduced electron carriers. Reduced electron carriers would be, say, our NADH, FADH2. It does produce a small amount of either ATP or GTP, as we'll see later. But a lot of the energy that is stripped off of our acetyl-CoA is stored in these reduced electron carriers. And our third stage of respiration is where these reduced electron carriers that are produced deposit those electrons into what is called the electron transport chain. So that energy is transferred, that energy is ultimately used to produce most of our ATP through a process called oxidative phosphorylation. And we'll discuss that a little later. So why is the citric acid cycle important? Well, it's a final common pathway for oxidation of fuel molecules. So in other words, these molecules like acetyl-CoA can be oxidized, and that energy, as we mentioned before, is conserved in our reduced electron carriers, primarily NADH. It also produces intermediates for biosynthesis. So your body does not just have to produce ATP, it also has to produce amino acids, things like nucleotides for DNA and RNA, porphyrin rings for, say, hemoglobin. And so some of the intermediates in the citric acid cycle can be used as the starting point for producing some of these very important biomolecules. So where does the citric acid cycle occur? Well, glycolysis, of course, occurs outside in the cytosol. The citric acid cycle occurs in the matrix of our mitochondria, which would be the inner part here. So our enzymes for the citric acid cycle are predominantly in the matrix. Now, as we mentioned, the citric acid cycle begins with acetyl-CoA, and if we recall, linking glycolysis to the citric acid cycle occurs as pyruvate is translocated into the matrix of the mitochondria, where the pyruvate dehydrogenase complex, through a multi-step process, converts pyruvate from a 3-carbon molecule to a 2-carbon molecule called acetyl-CoA. And this is a very high energy compound, as we discussed previously. So before we get into the actual steps of the citric acid cycle, let's just look at a very general overview. So here, this C2, that tells us that we have 2 carbons coming into the citric acid cycle. So this would represent our acetyl-CoA. Our acetyl-CoA is going to join with a 4-carbon molecule to form a 6-carbon molecule. And then as it passes around the cycle, it gets progressively decarboxylated. We're removing carbons in the form of carbon dioxide. Now, as we decarboxylate our molecule, we're also oxidizing it. In other words, oxidation is the loss of electrons, so we're removing electrons. And those electrons are stored in NADH, as you can see here, and also some FADH2. So these electrons won't be wasted. They'll go on to help produce ATP through oxidative phosphorylation. Now, there is a step where we actually phosphorylate ADP through what's called substrate level phosphorylation to produce either ATP or GTP. Most mammals, we produce GTP, but some lower life forms do produce ATP. So this is a very general overview of the citric acid cycle, and so we want to talk about that in a little more detail. So let's just review real quick that summary. For each acetyl-CoA that enters, we're going to produce 2 molecules of carbon dioxide through our decarboxylations. We have the coenzymes NAD+ and something else we'll talk about a little later, ubiquinone, which is sometimes abbreviated as Q, will be converted into NADH and QH2 or ubiquinol. So these are basically where we're storing some of those electrons that we're pulling off. We will produce one molecule of GTP as we phosphorylate GDP, or again, some organisms will produce ATP from ADP. And we also will produce or regenerate oxaloacetate, which, as we'll see in a minute, takes place in the first step of our citric acid cycle. So here's a general overview of the cycle. What I'd ask you to do is maybe pause the video and take a look at it. So here's where we're going to start, where our acetyl-CoA is going to come in, and our acetyl-CoA is going to combine with oxaloacetate, which we just talked about. So we're going to combine those two. The enzyme is called citrate synthase, which is going to take acetyl-CoA, the acetyl group, combine it with oxaloacetate to produce citrate, hence the citric acid cycle. So this is our first step, and then after that we're going to go all the way around the cycle until we end back up reproducing or regenerating our oxaloacetate, so we can go through another cycle of our citric acid cycle. So let's start by taking a look at this very first step, where we have oxaloacetate combining with our acetyl-CoA to form citrate, catalyzed by citrate synthase. So here is reaction 1, which we said was catalyzed by a citrate synthase. We take that acetyl-CoA, which was produced by the pyruvate dehydrogenase complex. We're going to combine that with oxaloacetate. Now, we have also talked about oxaloacetate when we talk about the gluconeogenesis pathway. That's one of our intermediates in that pathway. These will be combined in order to form citrate. Now, if we look under standard conditions, this has a very negative delta G standard. So this is a very exergonic reaction, because we have the acetyl-CoA, which is a thioester. We talked about that previously. And we're hydrolyzing that, or breaking that thioester bond, which will release a lot of energy. So this basically ensures this large release of free energy, ensures that the whole pathway, the whole citric acid cycle, will be exergonic and spontaneous, even if we have low concentrations of intermediates such as oxaloacetate. It will still be drawn or moved to the right. So let's just look briefly at the mechanism for citrate synthase. Now, we don't have to understand all the steps here, but essentially, if we look here we have our acetyl-CoA. The acetyl-CoA is reacted with a base in the active site in order to form a more reactive enol. This enol then attacks our oxaloacetate. So essentially, we're adding our 2-carbon acetyl group onto our oxaloacetate to form our 6-carbon intermediate here. And that's what you kind of see here. We don't need to worry about the names of this, but we have our 6-carbon intermediate. What I want you to notice here is that we still have that coenzyme A bonded. Remember, the hydrolysis of that thioester bond, or that thioester bond, releases a lot of energy. So we don't want to just cleave that right off the bat and waste that energy. So once we get our 6-carbon intermediate form, then we have a spontaneous hydrolysis of that thioester bond, releasing our 6-carbon citrate molecule here. So that the hydrolysis of the thioester bond occurs at the very end of our mechanism, so that free energy is coupled to the formation of that carbon-carbon bond in order to form our 6-carbon citrate molecule from the 2-carbon acetyl and 4-carbon oxaloacetate. So let's look at our second step. Here in our second step, we're going to take the citrate, which was just formed, and convert that into isocitrate. So we're going to undergo an isomerization reaction, and the enzyme that catalyzes this is called a aconitase. It's called a aconitase because the intermediate here is called a aconitase. And essentially, it converts this 6-carbon citrate, which is not chiral, so it has no stereo centers, into a chiral molecule, isocitrate. So we get a dehydration and then a rehydration. So we're removing water and then adding water back in order to get our isomerization. So we're essentially moving this alcohol group down from one carbon to another. This reaction is somewhat somewhat endergonic, at least under standard conditions. Under cell conditions, it's closer to zero. But the next reaction that we'll look at, reaction 3, is much more exergonic and will help pull this to the right and keep our cycle going in the exergonic spontaneous forward direction. So let's look at our third reaction. We're taking our isocitrate that we just produced via a conatase, and we're going to produce a molecule called alpha-ketoglutarate. The intermediate here is oxalosuccinate. We don't need to memorize the intermediates, just mostly the reactants and products, but there is an interesting step here. This is an oxidation step. The reaction that, the enzyme that catalyzes this is called isocitrate dehydrogenase. Dehydrogenases often catalyze oxidation reduction reactions, and that's true here. So we're oxidizing our isocitrate. We're removing 2 electrons and a hydrogen. You can see here 2 electrons and we're removing some hydrogens, and those are going to go to our 2 electrons and 1 hydrogen are going to bond to NAD+. We're releasing one of the hydrogens as a hydrogen ion, and we're forming our first reduced electron carrier. So that's a very important about reaction 3. Now this is a 2-step process. We have our oxidation, and then we also have a decarboxylation. So we're going to remove a CO2. So we're going to release a carbon dioxide. So we're removing that that carbon dioxide. So a couple of very important things about reaction 3. We produce our first reduced electron carrier. We also have our first decarboxylation reaction. So we're going from 6 carbons down to 5 carbons. And as we said in the previous one, this is fairly exergonic. And so that energy, a lot of energy is released. And a good bit of that energy is stored in that reduced electron carrier, NADH, which we can use through oxidative phosphorylation to produce more ATP. So let's move on and look at reaction 4. Here we take the alpha-ketoglutarate that we just produced, and we're going to produce a molecule called succinyl-CoA. This reaction is catalyzed by alpha-ketoglutarate dehydrogenase, or sometimes the alpha-ketoglutarate dehydrogenase complex. And it's a complex because it is a multi-part enzyme. It is actually very, very similar, or at least analogous, to the pyruvate dehydrogenase complex that we talked about as a precursor step to the citric acid cycle. So essentially what happens here, this enzyme has the same cofactors. It has an E1, E2, E3 portion. The E1 portion has TPP as its cofactor. And so the first step, as the alpha-ketoglutarate comes in, it's going to bind to the E1 portion, to the TPP, and we're going to get a decarboxylation. That intermediate is then going to get transferred to a coenzyme A. So we're going to form that succinyl-CoA. Instead of acetyl-CoA, we're forming succinyl-CoA. Now this process, we have to regenerate the enzyme, so those that lipoamide, which is going to be in our E2 portion, the reduced lipoamide is going to get oxidized, and those electrons will ultimately get transferred to NADH. So again, review that pyruvate dehydrogenase complex, and this is a very analogous step. It is also very exergonic. So a couple of important things about this reaction. Again, we're going to produce more of our reduced electron carrier NADH. We're going from 5 carbons to 4 carbons, so we have a decarboxylation. Hence, we have a dehydrogenase because this is a redox reaction as well. So, and it's very exergonic. So we're producing a succinyl-CoA, which is also a very high energy compound, just as acetyl-CoA is. So here's just a kind of a summary of what we talked about there, comparison rather, of the pyruvate dehydrogenase complex and the alpha-ketoglutarate dehydrogenase complex. They start with essentially the same reactants, and we produce very, very similar products The only difference, again, is instead of using pyruvate, we get alpha-ketoglutarate, and instead of forming an acetyl group, we form succinyl-CoA. So let's move on and look at reaction 5. Of course, we produce succinyl-CoA, which is the product of our last reaction, and we're going to convert succinyl-CoA into a molecule called succinate. The enzyme that catalyzes this reaction is succinyl-CoA synthetase. And here, a very important thing is this enzyme will also perform a substrate level phosphorylation. So if we look at this overall mechanism here, we have succinyl coenzyme A, and we're going to cleave off that coenzyme A. Now again, that's very exergonic, and what that does, that energy is coupled to adding a phosphate group onto our succinyl group. So adding a phosphate group, just directly a free phosphate, is very endergonic. That energy comes from the hydrolysis of that coenzyme A, that thioester, and it's coupled to adding a phosphate onto succinyl intermediate to form succinyl phosphate. Now that succinyl phosphate, the phosphate will then be transferred onto our enzyme. In the active site, there's a histidine which will get that phosphate transferred to it, and then that phosphate will get transferred to ADP to form ATP. Now again, it's also in humans and most mammals, it's GDP is going to get phosphorylated to form GTP. So as we see here, the potential energy stored in the thioester, this is our thioester right here, is used to form a nucleotide triphosphate. Again, most of ours is going to be GTP. So you can see how we conserve that energy as we go, and we use it to ultimately phosphorylate through what's called substrate level phosphorylation. So we talked about the intermediate that we form, that phosphate is transferred directly onto the enzyme, and then it's added to form GTP or ATP, depending on the organism. So let's keep moving on and look at reaction 6. Now we formed succinate, and we're going to take succinate and convert it into a molecule called fumarate. The enzyme that catalyzes this is called succinate dehydrogenase. So a dehydrogenase, we're probably going to undergo an oxidation reduction reaction. And those reactions are typically exergonic. So succinate dehydrogenase catalyzes the dehydrogenation of two saturated carbons to a double bond using the enzyme bound FAD. So essentially what we're doing there is we're taking off from succinate, our enzyme is going to strip off two hydrogens, which we can see here and here, and also two electrons, and we're going to transfer those electrons to FAD to form FADH2. Okay, we looked at this molecule when we talked about the pyruvate dehydrogenase complex, and this FAD is bound to the enzyme. It is covalently bonded, so it's not able to leave or diffuse out of the active site. So we form fumarate, which has a double bond. And again, here's our structure of FAD. Here's our reactive portion here, and you can see it's chemically bonded to our enzyme through this histidine. Now, another interesting thing here, I want to mention it here, but we'll talk more about it when we get to the oxidative phosphorylation, the electron transport chain. The electrons that are stored here, the FADH2, how do they get into the electron transport chain? Well, they will be added to a molecule called Q, coenzyme Q, also called ubiquinone, and those electrons are going to be transferred to this molecule to form the reduced form of coenzyme Q, and this coenzyme Q will take these electrons directly into the electron transport chain. So most of our electrons enter the electron transport chain through NADH, but this is a very interesting enzyme because it's part of the citric acid cycle, but this enzyme is also part of the electron transport chain, so it has two functions. So keep that in mind here, and we'll explain it more when we talk about the electron transport chain. We'll see this complex come back up. So succinate dehydrogenase is regulated. A little bit about its regulation. It's regulated or inhibited by a molecule called malonate, which is very structurally similar to succinate. So a couple of more steps. Reaction 7, we take our fumarate, which we generated, which has 1, 2, 3, 4 carbons. All right, we went from 6, now we're down to 4, and we're going to convert that into malate. The enzyme that catalyzes this is fumarase. And then our last reaction in the citric acid cycle, reaction 8, we're going to take malate, which we just produced, and we're going to convert that to oxaloacetate using malate dehydrogenase. So this is a dehydrogenase, so we're going to strip off a last 2 pair of electrons that we can, and we're going to add those 2 electrons. We're going to oxidize malate. We're going to add those electrons along with the hydrogen, so a hydride onto NAD+ to form NADH. So it gives us just a little bit more energy to add to our electron transport chain. Now, under standard conditions, this is fairly endergonic, but of course, as we looked at before, our very first reaction, which would come next in our cycle, is very exergonic. So that keeps even with this having a fairly high delta G value, the overall cycle is still very exergonic and spontaneous. So again, just a quick review. We started with our acetyl-CoA, our 2-carbon, converted that to citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, back to oxaloacetate, so we can go through it again. Now we want to be sure we appreciate each of our enzymes that catalyze these steps. We want to be sure what these enzymes do if we have a dehydrogenation or if we're forming a reduced electron carrier, where we have our decarboxylation steps, and any other interesting points that we talked about in our lecture. Now, just a couple of more things about the citric acid cycle. I want to show you this slide just to point out our delta G values, kind of like we did for glycolysis. Under standard conditions, so under cellular conditions, we have a very negative delta G for our first step. Most of our other reactions are very close to 0, which means they're not very endergonic. They could be reversible, but because we have a few very exergonic steps, then this process, our overall citric acid cycle, will always be in the forward and spontaneous direction. Now, the difference between, of course, delta G standard and delta G non-standards is typically related to concentrations. So as our concentrations are very different in the cell than they are under standard conditions, that's why we have those differences. And just to revisit one last thing that we talked about at the very beginning, the citric acid cycle is key in giving us those reduced electron carriers that we can use to make our ATP through oxidative phosphorylation, but it also is an anaplerotic pathway. In other words, it produces intermediates which can be used to make other biomolecules in other pathways. For example, oxaloacetate, which is produced in our citric acid cycle, can go through a different pathway to produce aspartate and then other amino acids. So we don't have to build amino acids from scratch. We can start with these building blocks. Citrate is a building block for certain fatty acids and sterols. Alpha-ketoglutarate, again, can be used to make other amino acids. And succinyl-CoA can actually be used to make our heme group, our porphyrin ring. So this is a very important pathway, not just in energy production, but also producing intermediates that can be used for other pathways.